And good afternoon to everybody, and welcome to this another in series of AHDB webinars, this time on the impact of research on blackleg management. For those that don't know me, I'm Graham Bannister. I'm one of the knowledge exchange team in the potato sector. I'm now going to use a very long word, which I'm not going to use again, but hopefully the other guys who are going to much more knowledgeable than I will pronounce it properly. Pectobacterium atrosepticum, and that is the last time I will say that. The pathogen responsible for potato blackleg and soft rot of potato is one of the most economically damaging bacterial plant pathogens for the UK potato industry. Today, we are very lucky to have a distinguished lineup of speakers who will present some of the impacts from an AHDB and Scottish Government funded project looking at how improved seed management can minimise losses due to blackleg. If you could move to the next slide, Christian. So we'll start with Ian Toth, Senior Scientist and Director of Scotland's Plant Health Centre, James Hutton Institute. Then we'll go into Greig Cahill, Bacteriologist from SASA, Glyn Harper, Senior Scientist from AHDB's Sutton Bridge Crop Storage Research, Stuart Wale, Potato Consultant and Research from SAC Consulting, John Elphinstone, Plant Bacteriologist from Fera Science Ltd, and then Ian Toth will round up and talk about a newer project which we are keen for you to know about. We'll wrap up with some questions and answers at the end. If we do get questions in beforehand, we will try and maybe pick up a couple in between speakers, but basically we will end it to the end. Before I pass over to Ian, there is a bit of housekeeping that I need to keep you informed of. Firstly, everybody, all attendees are on mute. But if you do want to ask questions, please do so and put them in through the questions tab, which is to the right hand side of your screen. We will hope to finish within the hour and a quarter we've got, so we're aiming to be done by 5:45. If we run out of time and we haven't got all the questions answered, we will get back to people individually via email, but hopefully we will have done that. We have got basis and then Roso points available for today. If you just put in normal information that you would put if you were actually at an event in person with a piece of paper in front of you, if you put all that into the question lines, Christian in the background will deal with that for you and apply for the points. It's worth pointing out that that information won't be shared with anybody else, only the people that need to know to get you the points. We also are recording this webinar, as we do all of them. This can be seen at a later date if you have to disappear or go and do something else by visiting the AHDB's website and go to the events page where all our previous webinars are recorded, which is a useful place to find them. We also have Twitter. Obviously, a lot of people will be using that. So if you're on Twitter, please tweet away using the hashtag Blackleg. And basically, don't forget, keep the questions coming in, because like I said, we've got a great lineup of speakers here today, some great knowledge about the topic. So keep the questions coming in. And with that, I will pass over to Ian.
So thank you, Graham, and welcome everybody. I'm going to chair the rest of the meeting. And first thing to say is thank you very much indeed for coming along today and taking your time. It's a real shame that we can't see you in person, and I personally really enjoy coming to grower events and meeting different people and discussing issues of blackleg and other things. Because of COVID, of course, it means we can't do that today. But if there's a silver lining, I think that is that a lot of people that aren't always able to come to face-to-face events do make an effort to come to this, because it's just an hour and a quarter in this case. So welcome to them and welcome to everybody that's made the effort to come here today. So as a way of introduction, I just want to say that today we're going to give you a very brief overview of some results to come out of two different projects. They've got the following scientific aspects: people from SAC Consulting, SASA, Fera, Sutton Bridge, Crop Storage Research in the James Hutton Institute, and we're going to be speaking today. But all the projects that we do in the past and the ones coming up in the future have got significant involvement of industry, and we really do appreciate the industry support that we get for this and the knowledge that we can get from industry. And we try and share the knowledge together to make sure that we come up with some practical differences. And then finally on this slide, I want to thank AHDB and Scottish Government for recognising the importance of work on blackleg and for funding it. Next slide, please, thanks. We have a number of fact sheets, I think about 5 in all, that we've attached that you can get access to today. These are just an example of 3 of them, so please do take the time to download them and have a read at them, particularly the new Blackleg Essential Facts leaflet, which we've only produced in the last couple of months, which really highlights a lot of the things that we know about blackleg. And this is, we hope, a useful document for you to look at, but more than that, we hope that you'll be able to find things and ask questions on some of these points, which we hope you can do this afternoon. And next slide, please. And then final slide before we start the talks. We're going to have these short talks, just 5 minutes each, to give you an idea about some of the work we've been doing, for about 45 minutes in all. And then we'll have a poll question at the start of every talk, which we'll look at the end in the discussion. And we'll have a question and answer session and a discussion for about 30 minutes at the end. So the speakers are on the bottom. I'm sure you know most of those faces, if not all. I particularly want to thank Sonia, who's not speaking, but has been very heavily involved in managing these blackleg projects and in organising today's webinar, along with these guys from AHDB. And thanks very much to AHDB, not only for funding the projects, but for hosting today. And thanks particularly to Christian and David, who are the unsung heroes in the background that do all the technical stuff that we very rarely see. So thanks a lot to you guys. Okay, so next slide, please. So thanks again for coming. I'm going to stop there and I'm going to pass over to Greig to speak about testing minitubers for Pectobacterium. Thanks, Greig.
Thanks very much, Ian, and good afternoon to everyone. So I'm Greig Cahill, I'm bacteriologist at SASA, and this is a small part of one of the projects that Ian mentioned that's funded by the AHDB. And what we're looking at is basically sources of initial infection of Pectobacterium atrosepticum. So we know that the infection is mainly seed driven, and that the more bacteria you have in seed potatoes, the more infection you're likely to get the next year over the whole seed potato system. However, that doesn't explain where the initial infection comes from, and that's something we're trying to look at the moment. So next slide, please. First of all, though, we're going to have a poll. So the question of the poll is, have you ever seen blackleg in first generation crops? So if you're growing PB1 crops from minitubers, have you ever had any blackleg in your crop at all? So we'll maybe give 30 seconds or so just to answer that question.
Yep, we're getting a few in here. I'll leave it another 5-10 seconds or so. Just to let everyone know, we're going to be showing the results of the polls at the end of the webinar as well, because we've got a few coming throughout, but then we'll discuss them at the end through the Q&A as well. I'm going to close it now. I think that's quite a few people voted. So over to you again.
Okay, thank you. Okay, thanks for everyone who's voted there. So just as a bit of a recap, if we could move on to the next slide, please. So the microplant production happens at SASA under controlled conditions in the nuclear stock initiation unit. So all seed potatoes in Scotland originate from microplants at SASA, and these are all tested for all sorts of potato diseases, such as the viruses listed here, any pathogenic bacteria such as the ring rot and brown rot, and of course Dicea and Pectobacterium that we're interested in at the moment. So once they're grown up and multiplied, they go on to the minituber producers. If we can go on to the next slide again, please. So these are grown again in controlled conditions. So the microplants are, further cuttings are made at the microplants, and then they're bulked up in either using hydroponics, which you can see on the right, or aeroponics, should I say, or they're grown in peat, as seen on the left, in these glasshouses under controlled conditions. And these facilities are obviously they're regularly inspected by ARE staff to make sure that everything is disease-free, is not showing any symptoms, and biosecurity is being adhered to. So if we can move on to the next slide, please. So despite all these measures that we take, starting from clean cuttings, growing minitubers under controlled conditions, we still find that we're getting Pectobacterium atrosepticum infection in tubers grown from pre-B1 stock. So if you see the table on the left here, it shows you the percentage of stocks tested over a 4-year period that we actually found Pectobacterium atrosepticum in. So on average, we're looking at about 14% of these crops from first grown seed tubers actually have picked up an infection somewhere. And if we look at the table on the right, this is a slightly different method in where we test core and peel samples, but we use enrichment techniques, which means we can find really small amounts of the bacteria. And if you look at these results here, we can see we can find 18% of the stocks tested had Pectobacterium atrosepticum, and using the peel, we can find over 30% of these stocks. So what we're seeing here is we're coming from completely clean microplants, completely clean minitubers. Well, clean minitubers we assume to, and that's what I'm going to show next when we test them. And we still find that the PB1 crops have quite a large infection of Pectobacterium. And obviously this, once it's into the seed production system, it multiplies over the years, producing more and more blackleg. So to give you the results of this testing we did, can we move on next slide, please? So this is a testing program with over 3 years where we tested minitubers from all the producers in Scotland. We tested a total of 107 varieties or clones, which amounted to 1,500 subsamples, which were 20 tuber subsamples each. So in total, we tested approximately 30,000 tubers over the 3 years. And we didn't detect any Pectobacterium atrosepticum at all in any of these. And this was using the enrichment technique as well. So we're confident that if there was even a very small infection of Pectobacterium, we would have found it. We also, alongside blackleg pathogen in Scotland, we tested for the other potato pathogens, Pectobacterium resiliense and Pectobacterium parmentieri, which is previously wasabi, which we didn't find any of either. And we also tested for Dicea species, which we didn't find any. So based on this testing, we're pretty confident that we can say that the minitubers are unlikely to be the source of the initial Pectobacterium infection in field-grown crops. So that would leave the question is, where does the initial infection come from? And that's something we are looking at the moment in our new project that Ian will describe later on at the close of this webinar. But ideas, if it's not the seed tuber, is that it could be through rain splash, through aerosols, it could be through irrigation water, or indeed through weed rhizospheres as well. So there are some other likely methods of transmission, but we're pretty sure now that minitubers aren't harbouring any Pectobacterium at all, like in the first generation seed. And that is the end of my talk. And I think Ian is going to pop up again and see if there's any questions.
I am indeed. Thank you very much, Greg. Could I just remind people that if you do have questions, you can ask them at any time through these talks. You don't need to wait till the question and answer session. Just we can't promise to get to it straight away, but just feel free to ask one if something springs to mind. There's one question coming up here which I can ask. What's the source of Pectobacterium that contaminates first generation crops planted from minitubers?
That's the good question. That's that little diagram might have answered your question after you'd asked it, but yeah, we're looking at various ideas which could be aerosol transmission. So aerosols can travel in the atmosphere for miles. So if you have, say, a field that's got quite heavily, like in a heavy rain shower, they could create aerosols which trap this bacteria in the aerosols, which then get lifted up into the atmosphere, and then they could travel for miles before they get deposited on P-basic seed. Although it's not going to cause infection that year, it could get into the soil and then get into the roots and through there into the tubers. There's also potential of weeds harbouring the Pectobacterium. Well, Ian and his team have done some work on that, but we don't think there's any likely weeds that grow in potato fields that are likely, so we're not sure about that. Ian's also looking at nematodes as well as potential harbouring the infection. So there's various methods that we are currently looking at, and that's one of the main questions in the next lot of research that we're doing is where does this initial infection come from? And if we can actually find out where it comes from, can we stop it or at least postpone it a couple of generations to ensure the health of the seed?
Thank you very much, Greg. I think in the interest of time, we'll move on. People are asking questions, and I think we'll come back to those as we go through the other talks. So thank you very much, and we'll move on to Glyn Harper. Thanks, Glyn.
Good afternoon, everybody. So this part of the talk is concerned with the management of soft rock during storage. So there's a component of the project involved in how we could satisfactorily manage soft rock during storage. I think, are we expecting a poll question? Oh, so the poll question is, do you have positive ventilation in your store? And the reason for that might become obvious between mine and Stuart's talk.
So we're getting a few votes in again. I'll leave it for another 10, 15 seconds or so.
All right, thanks for that, Christian.
I think it's starting to slow down now, so I'll close it now and pass back to you.
Yeah, okay. So this is the pathologist disease triangle, which has the three components that are required for a disease to occur. So the first is a host that's susceptible to a pathogen, and in that case, this is potato. Unfortunately, none of the varieties of potato that are typically cultivated have any resistance at all, really, to Pectobacterium. The second part is a pathogen that can infect the host, and here we've got Pectobacterium species. And the third is the environment, and specifically here I'm talking about the microenvironment of within a store. Next slide, please. So the trial, we originally conceived the trial, we thought we would be able to attack the pathogen component of the triangle using two different techniques. One is pulsed UV light, and the second is bacteriophages. Bacteriophages are viruses that infect that bacteria, specifically infect bacterium. So these work fantastically well in very highly controlled environments, so in petri dishes, in the lab, and bacteriophages are actually used commercially after washed potatoes. But here we had the idea that we would use them at the start of storage and try to see if we could then control the levels of bacteria during storage. Unfortunately, after 2 years, we found we couldn't really find results that were convincing. And that's not to say that they don't work. It's just that I don't think we could get a methodology that would allow them to work for various reasons. So maybe in a different system, maybe in the field, for example, or where there's a bit more control, I think there's still potential technologies for controlling the pathogen. So then we moved on to the environment. So next slide, please. And we set up what we thought would be a trial just to show that, or to see if the control of these three factors, or two of the factors within the store, would be useful or critical in controlling soft rock disease development. And specifically, that's drying and ventilation and storage temperature. Condensation control is absolutely vital as well. Next slide, please. Just before I go on to that, I just want to put in a word for sampling before storage. It's no good having really good storage facilities and really good control if the amount of disease that you're putting in is simply uncontrollable. But this is I'm sure all growers would do this, but it's actually quantitatively assessing the disease of their stock before it goes into the boxes or bulk, and then using that assessment to gauge the levels of control that are required. So anything more than 1% rots, bacterial soft rots, is really difficult to control. Anything over 2% is essentially uncontrollable. So you need to know that before storing. Next slide, please. This is the actual trial. So on the right hand side you can see a plastic box which is slightly smaller scale than the boxes you saw in the previous slide, but it's still adequate for us to work with. These little sandwich boxes have got fans in them that are operating at the same ventilation rate that you would see in a store, and we can store them at various temperatures, so 3.5 or 15 degrees. The ventilation regimes we used were no ventilation or normal store ventilation. And the tubers were artificially inoculated because one of the problems that we had with the previous trial, we thought, is just the level of natural variation of soft rot within a stock. It really confuses, makes analysis very difficult. I'm not going to show you the results here, but just the conclusions that we could draw from those results. And there are three really. And the first is that immediate drying of the seed tubers by ventilation reduced inoculum numbers. So these bacteria need water really to survive well. And if you take that away, they will die off. Increased tuber loading. If you don't adequately ventilate, you can't reverse it to later date, especially under warm conditions. So that is, the bacteria seem to be able to either multiply or increase in number, particularly under warm conditions, even with ventilation. And the third is that ventilation does not eliminate all Pectobacterium, atrosepticum in this case, from tubers, but does decrease risk over the storage period. And that's because many of the bacteria will actually be internal within the tuber, in the vascular system, for example. Next slide, please. Temperature is obviously a key control. I didn't really bring it through from that last trial, that there was a difference between 3.5 degrees and 15 degrees, as you might expect. And that's really shown in that the graph on the top left. At the bottom, the x-axis is temperature, and it goes from 0 to 20, and on the y-axis is the time that it takes for the bacteria to multiply tenfold. So if you just look at 15 degrees, it's taking just under a day to multiply tenfold. If you look at about 6 or 7 degrees, it's taking around about 5 days to multiply tenfold. During that time, had those bacteria been kept at 15 degrees, there would be 100,000 times. So that multiplication is huge, and it shows you why temperature is so critical, and why it's so important to get the temperature, the heat out of the store potatoes at store loading. The bottom right graph is actually an ambient store temperature logger in one color, and humidity at the top, rolling along the top. And you can see before 1st of November, when the fridge was turned on, you can see fluctuations of 4 perhaps 4 or 5 degrees. And those are events where the store temperature, obviously the store temperature is not being controlled except through ambient, and it's periods where condensation could be occurring. And condensation is a clear risk factor for soft drop development. Next slide, please. These are 2 documents, and actually you saw them at the beginning. The first one on the left-hand side is the AHDB Store Manager's Guide. Most of what I've talked to you about is actually store management, so ventilation, temperature control, making sure the area is being able to go through all the potatoes as effectively as possible. And this guide is really concise, really good information about how to manage those stores. The right-hand document is a really nice government document produced by the Scottish Government organisations, and it's that integrated pest management type of control with all the various factors through the life cycle of the potato cultivation, which you can impact to reduce the risk. So the final project report from this project, it will shortly be available, and in that we will be able to highlight what we consider best practice storage advice. And that's the end of my talk. Thank you.
Many thanks, Glyn. In the interest of time, we are going to go straight into Stuart's talk. There is a storage question, but we'll save that for later if you don't mind. So, Stuart, if you wouldn't mind carrying on, please.
Good afternoon, everyone. I'm going to take on the storage story from the interstore period that Glyn has talked about. I think the next slide will show my poll question. And this is really, do you attempt to clean your grader? I don't mean take away tubers that have left from the grading line, left on the grading line. I mean physically clean the soil or spray the grading line with chemical, a disinfectant. 3 choices between stocks. Do you do it between stocks, all stocks or most stocks, or do you do it after a disease stock, or do you do nothing at all? So if you could vote on one of those options.
Still getting a few more in, so I'll leave it a little bit longer. To those who've joined late, we're going to be showing the results of all these polls at the end, just before the question as well. Yep, it's slowing down now, so I'm going to close it and pass it back to you.
Thank you, Christian. Well, it's perhaps obvious to us all that there is a strong link between how we store seed potatoes and the incidence of blackleg developing in the field. Storage and handling can have a beneficial effect. We know that we can reduce the inoculum of PBA or Pectobacterium atrosepticum on the tuber surface, or it can have a negative effect. And this project, or the project just finished, has attempted to quantify some of the effects on during the storage period. Like Glyn, I want to emphasize the triangle, in this case, a 3-way process between the bacteria, temperature, and tuber wetness. The part of the project was to do a literature review on the effect of the environment on Pectobacterium, and that's been published, I believe, but certainly will be part of the final report. And the quotation top right, the rate of pathogen multiplication at infection sites is affected by temperature, humidity, and oxygen potential. So to illustrate that slightly, I've got a graph below, similar to the one that Glyn showed you, but with flatter lines. The purple is relative humidity and is on the left-hand axis, and the red line is the temperature on the right-hand axis. And this is a grower who warmed his potatoes during grading and then dropped the temperature down to a very uniform level Despite the purple line being very high, probably nearly 100% or close to 100%, which of course you need to avoid shrinkage. You might think there's a risk of condensation at 100% humidity, but only where the temperature fluctuates. And in this particular example, the fluctuation was absolutely minimal and the condensation risk was negligible. Next slide, please. Following on from what Glyn said, this is a case where the stock, this is from the project where the stock had rotting going into store. It was ventilated up until early February, I think, and then, but it didn't stop the rotting in the stock. And then the stock was graded on the 22nd of March. And the graph is on the left-hand side is the log scale. So 1 equals 10, 2 equals 100, 3 equals 1,000, and so on. So increasingly more severe contamination. And on the left-hand side, the bar is pre-grading, had about 300 bacteria per tuber. And after grading, the contamination, the spread from the bacteria was so great that it went up to, on the right, the very far right-hand side, up to nearly 20 million bacteria per gram of tissue. So a very significant effect of grading a rotting stock. Next slide, please. So I want to talk a little bit about the grader. We tried to look at, should I say, at cleaning a grader, which is an extremely difficult task. We were set the challenge over a lunchtime to try and clean a grader, and someone was physically taking soil off, and my colleague Inez Jessamine was spraying it with peracetic acid. To do that on a fairly complicated piece of equipment in half an hour of lunch break was nearly impossible. And our conclusion was that during a busy grading period, misting with a 3% solution of peracetic acid after a diseased stock may be the best option for cleaning that there is available. It doesn't have to be wetting, just misting. It was difficult to follow, to determine whether cleaning a grader had any great effect, but our view is that after a diseased stock, it probably is worth doing. Next slide, please. Now, during the grading process, like the rest of the storage period, we want to avoid moisture, particularly as you're going to put those potatoes, these seed potatoes, into a bag or a box. But there are 3 sources of moisture we identified during the research program. There is the physical action of the grader, and you'll see top right, a step grader or jump grader. And on next to that, the tubers are showing expressed moisture and some damage. So that's one source of moisture on the tubers at grading. The second is some growers are using a light spray of water to reduce dust. That definitely is adding to the moisture content of the tubers. And that's in the middle on the left-hand side. And then if you look at the middle on the right-hand side, there is this dry steam or pressurized water, which is not wetting the tubers, which is a much better form of dust control, much better than dust extraction, for example, much better than spraying liquid water on. Many of the PB growers we looked at have that system. But if you put water on to reduce dust, that's also a negative. And finally, as you know, some people will treat seed tubers with a fungicide, and that's putting on 1 to 2 litres per tonne of moisture. So then that, next slide, please. So then that goes into, after grading, into a bag or a box. And we looked at we tried to evaluate post-grading ventilation of bags and boxes. Now the box is relatively simple to do because most people have positive ventilation systems they can use to dry out a box, and such as on the right hand side there. But the bag, which is the main system of transport for seed potatoes is very difficult to dry out the moisture in. Even vented bags, the air movement through the tubers within the bag is very limited. The obvious thing to do is to limit time in the bag to a minimum, but from experience, it may be after grading something between 3 and 7 and possibly up to 4 weeks on occasions where the bags sit without being moved. So clearly limiting time in the bag to a minimum is something you can do. Boxes, some people use boxes to transport seed potatoes, but of course there's the cost of the box to be considered when you do that. Next slide, please. So we did some experimentation to see whether putting tubers into bags or boxes made a difference. And the results I'm showing you here from the project suggest that putting them in a bag at least keeps the level of bacterial contamination the same or is greater. But I have to say the results over the 3 years of the project were were not consistent, so I can't hand on the heart say that bags are necessarily a bad thing, but intuitively that seems to be something we should consider. Next slide, please. We also looked a little bit at transport of bags and boxes and subsequent handling. And this particular example is the graph of humidity and temperature as before. And you can see where the black arrows are pointing during transport, small changes in tuber temperature occurred. These were fairly small and probably wouldn't have much effect on in causing condensation. But nonetheless, there is always a risk when you transport potatoes that you can get condensation and increase the bacterial multiplication. In the first case, on the left-hand side, the grower put the potatoes, decanted them from the bag into a box and put them straight into a cold store and kept the temperature uniform until just at planting time. And on the right-hand side, right-hand graph, it was also in a cool store, and the grower in this case took the potatoes out of the store some days before planting, and you can see a very big jump in temperatures. This is 2018 when the air was very warm outside. Again, there's a risk there of condensation occurring before planting. Next slide, please. So that gives you a little bit of a flavour of some of the storage work we did. The final report, as Glyn said, will be out fairly shortly. But I wanted to finally mention that to reduce blackleg in the field, you had to take some agronomy actions. First of all, you want to avoid desprouting, as shown on the left-hand picture. That is, we were following, some of the growers we followed did have examples of where desprouting resulted in increased blackleg. Similarly, we also had examples where growers had rhizoctonia in the field, and that also increased blackleg in the field. So controlling the temperature in the store can limit the sprouting, and that makes it less risk of blackleg, and also putting on seed tuber fungicides when they're required for controlling rhizoctonia is a good thing. Good practice. That's the end of my talk.
Thank you very much, Stuart. Again, in the interest of time, I think we'll move straight on and come back to questions after. So, John, if we could go to you, please.
Thanks, Ian. Good afternoon, everybody. My slides, I just wanted to show some data on how we've been genetically differentiating strains of Pectobacterium atrosepticum to see whether we could study their spread during basic crop multiplication. Start with a poll question. is it possible to see significant different levels of blackleg when the same seed stock is planted in two locations?
Getting some quick votes in this time, so I'll close it in the next 5 seconds or so. Cool, I'm going to close it now, so back to you.
Thank you. Next slide, please. So we've used a couple of methods to type genetic diversity within Pectobacterium atrosepticum PBA strains, and we've tried to use these methods to compare to monitor the spread of these various strains in the field, and we wanted to see whether we could use this sort of approach to identify possible sources and pathways of contamination with PBA coming into first field generation crops. Next slide, please. In the previous projects, we've looked at something called VNTR profiling, and we've reported quite extensively on this. We found 18 different strains of PBA with different VNTR profiles. This data is showing what happens when you look at 8 different seed stocks all growing in the same field, and you type the you isolate and identify PBA, and you type them from blackleg plants or from the progeny tubers after harvest. And we could see that the PBA from the blackleg plants was less diverse, so only 2 strains in this case, causing the blackleg in all of the different varieties and seed grades, irrespective of where the seed stocks originated. And then when we looked at the progeny tubers after harvest, we could see that they were picking up additional strains. So you see slightly more color variation in the pie charts showing the distribution of the different strains. Next slide, please. VNTR typing, it's only possible to detect to type small numbers of strains. We looked at 300 in our studies because we have to isolate the organism first and identify PBA before we can actually do the typing. At the James Hutton Institute, there's been developing a new system of typing called DNA barcoding, which uses specific markers that are only found in Pectobacterium atrosepticum. And with a single marker, it's been possible to identify 3 different main genotypes, which are PBA specific. And the benefit of this kind of approach is that it can be automated. It doesn't need isolation of the organism first. You extract DNA and you look at the relative abundance of all the PBA strains present in the sample. Next slide, please. So we compared with our 300 test isolates that we'd saved from previous projects, originating from 3 different farms, 1 in Yorkshire and 2 in Scotland, in 3 different years. And we saw that there was not really any agreement between the VNTA profile and the barcode type. So the 3 main barcodes, 1, 2, and 3, had a mixture of all different VNTA types in them. But what the main finding was that we could find all 3 of these barcodes on all 3 farms of origin in all 3 years. So using this single marker approach, we're not getting really sufficient diversity to be able to use this kind of approach for source tracing. Next slide, please. What we could show was that with either the barcode typing or the VNTR typing was that when we planted the same minituber stock in the 3 different locations, then we got different patterns of contamination. Within the first field generation there on the left, you see at farm 1, we got barcode 1 and 3 and VNTR 9, but at farm 2, barcode 1 and VNTR 1, and at farm 3, we didn't get any infection at all. So this is going back to what Greg was saying at the start, that we don't think the source of infection here is from the minitubers. It's more likely to be coming from the local environment. And then you see in subsequent years of multiplying the same stock, then you get increasing numbers of strains coming in. But again, the patterns are different in the different locations. So this is a useful way of tracing, source tracing PBA coming into first generation crops. With additional barcode markers, we should be able to extend the amount of diversity we can find, and therefore use it more effectively for this source tracing purposes. The last slide, please. So just to conclude, PBA contamination appears to be site-specific in the early generations of seed multiplication. The VNTR analysis was not suitable. It was too expensive. It can only be applied on small numbers of strains. Also, it wasn't suitable for mass source tracing. But barcoding with multiple barcodes seems to be the way to go, and I think that will be the approach taken in the new project, which Ian will talk about now. Thank you very much.
Thank you very much, John, and we'll move on to the next one, which is me. I'm going to talk about a new project called the Decision Support Tool for Blackleg Disease, or DESBL. And this is a project that has a number of academic and industry partners and is about £2 million worth of funding. So it's a major project. It's in the region of 20 times bigger than some of the other projects that we've done. So we really hope to get some good data out of this over the next 3 years. It's funded by BBSRC, NERC, and Scottish Government and DEFRA. So we're looking really at a different type of funding in terms of some of these funders to really try and make a big impact with Blackleg. Next slide, please. The project originates from 2 main pieces of work that we've been doing, both within the projects we're going to talk about and within bigger Scottish funding work. The first one is the fact that if we take minitubers that we show are completely clean of the pathogen, and we plant them in the field and we over irrigate. I know you wouldn't normally irrigate these seed stocks, but we wanted to see what effect irrigation had. And we were actually amazed to find that in some of these fields, and this was repeated over 3 years, we were getting up to 7% blackleg in a first generation crop. So the irrigation's doing something that's leading to that blackleg disease, not just contamination, but blackleg disease. We did another piece of work looking at free-living nematodes, and it's been suggested that free-living nematodes could be involved in causing root damage, thereby allowing bacteria to get more easily into the roots. So we did an experiment where we included free-living nematode and Pectobacterium in the soil with growing plants, and we saw up to 100 times more bacteria in the stems of potato plants than without nematodes. So it seems from that initial work that nematodes are making quite a big difference to allowing bacteria to get inside the plant. Next slide, please. So the first part of this new project is to look at irrigation, and we're particularly focusing not on the tubers because we know they're important for causing blackleg disease once they're contaminated, but this is looking purely at the root systems. We want to look at irrigation, different levels of irrigation, and we know that for ware crops, particularly in England and Wales, people tend to irrigate well, sometimes over-irrigate, to try to reduce the incidence of common scab, which often forms in dry soils. So we want to see if we can find a sweet spot so that we can minimise common scab and at the same time try to reduce the incidence of blackleg. We also want to see whether irrigation causes an increase in free-living nematodes, possibly moving up from deeper in the soil to around the root systems. And finally, we're working with Bayer Crop Science and their Vellum Prime nematicide to see whether adding a nematicide at certain times during production can reduce the nematodes and actually have an impact on blackleg in the field. So in this case, we're using a nematicide to control a bacterial disease. Next slide, please. So as well as irrigation, we want to look at the effect of cover crops in reducing or increasing numbers of Pectobacterium in the soil. We know that cover crops are becoming very popular because they play an important role in soil nutrition and quality, etc. But actually we know nothing about what happens in terms of Pectobacterium numbers. Do some cover crops increase Pectobacterium in the soil? Do others reduce it? We just don't know. So what we'd like to do is to look in detail at the amount of Pectobacterium that we get on the root systems of growing cover crops to see whether we can identify cover crops that increase or decrease Pectobacterium just prior to planting of your potato crop. And the idea here is that we might also find antagonistic bacteria that we can use to identify and develop a biocontrol agent that could then be added to a potato stock to try to bring down Pectobacterium levels. We also want to look at the role of aerosols and insects on the spread of Pectobacterium to plants, and that's something that we'll be doing within the project as well. Next slide, please. So finally, we have lots of amazing data that we can start to correlate with the incidence of blackleg disease. We have satellite data on soil moisture, on leaf wetness. We have data on agronomic values. We have data on climate change, and we have data on free-living nematode occurrence across the whole of the UK within 10-kilometre patches, and the agronomic data that goes with it. And with all this data, what we'd like to do is try to correlate it with the incidence of blackleg in different parts of the UK and over different years. And so we're going to put all this information into a model to see whether we can pull out certain factors that make the biggest difference, and hopefully, ultimately, to develop a decision support tool that you can use. We're working particularly with AHDB on this project to do knowledge exchange and communication, and really this is one of the first ones that we've done. The project started on the 1st of October, but we hope to do lots more KE events where we can tell you more about this work, more about the decision support tool, particularly as we start to develop it. And then we want to do an assessment of how effective that decision support tool is and uptake is. Okay, so next slide, please. So just to finish off, the practical outcomes we hope to get from the project are several-fold. First of all, can we improve cover crop management with blackleg in mind? Can we improve irrigation management? Could we, for example, identify the number and the species of free-living nematodes in soil and use that as a forecast for possible blackleg disease? Can we cover high-grade seed to protect them from aerosols and insects? Do we need to do that? And then can we develop new biocontrol agents? And finally, can we put all this together along with the modelling work to develop a decision support tool? And if only one of these comes off at the end of the project, we hope that it will make a difference to blackleg control. And then finally, we have a blackleg Twitter name. I think they call it a handle. I'm never quite sure. Blackleg space hub. And we also have a new website, blackleghub.ac.uk, if you want more information. Thank you, Christian. That's the end of that talk. So we've just given you a quick, actually about 50 minutes of talks. We hope that you've got something out of it. It all seems to have happened very quickly, but I think now in the last 20 minutes, we just want to go through the polls and then look at 1 or 2 questions. So if we can take the first results of the first poll, please.
No problem.
So have you seen blackleg in your first generation crops? Almost 40% of people have. So over to you, Greg. Do you want to comment on that poll?
Yeah, that is quite an interesting result. I suppose it's not borne out in the inspections, which are obviously just a small snapshot in time of when the inspection goes on. So it is interesting because generally, up until sort of maybe 5 or 6 years ago, when we did a project on blackleg, which we saw transmission in the first generation, we didn't really think there was much blackleg going on in PB1 crops. So that is interesting to show, basically showing that a crop can get infected and show symptoms within the first growing season. So that could tie in as well with the VNTR testing that John can do, that we could actually, if we could get hold of some of these crops, we could try and elucidate where this PB is coming from, causing the blackleg.
Okay, thank you very much indeed. If we could go to the next poll. Okay, so do you have positive ventilation in your store? 80% said yes. Could we go to you, Stuart, for to Glyn on a comment on this?
Yeah, sure. I'm sure Stuart will have a comment as well. I think that's slightly higher than I was predicting, but that's really positive news, actually. That obviously positive ventilation is a much more effective way of moving air through the store, particularly through the potatoes, compared to a more passive ventilation style, overhead throw or something. So I think that's a really encouraging result. Nice to know actually how many stores that relates to, but no, I think that's highly encouraging. Probably reflects the go-ahead nature of the people that attend these webinars actually as well.
Okay, thank you very much. On to the third poll. To you, Stuart.
So the results are showing 53% attempt to clean the grader between stocks, 26% after a disease stock, and 21% neither. It is a very difficult thing to do. I think I've blandly just said that you should be able to clean your grader, but having tried it, I can well believe how difficult it is. And I have been to some growers who never touch it between one season and the next. That's very encouraging.
Okay, thank you. And final poll. Is it possible to see significantly different levels of blackleg when the same seed stock is planted in 2 locations? John, over to you.
Fairly conclusive result there. It doesn't surprise me. I think what this is telling us is that a low seed is often blamed as the source of Pectobacterium causing blackleg. It's not the only factor by any means, and that many other factors can, which are more location specific, can come into play. And this is what I was trying to get across in my short talk there this afternoon.
Okay, thank you very much. So we're on to a question and answer session now, and thanks to all the people that have sent in questions, and we're going to start going through them. I'm not going to go in any order because there's a long list. I'll just get to them as I do, and we can pass them on to people. We have about 15 minutes left. So I thought that the first question is, have you thought about testing soil and weeds in fields destined for pre-basic 1 before they ever have them? Maybe that's one for John.
Sorry, I was fiddling with my camera. Could you repeat that again?
Have you thought about testing soil and weeds in fields destined for PB1 before they ever have them?
Yeah, we did this actually as part of this project and were unsuccessful in detecting PBA in weeds or soil before planting in the locations that we chose to do the experiments. So if it's there in very hard to find populations, very low populations.
Okay, thank you, John. There's a question
I would just like
here.
to comment as well. There's currently there's currently a Ufresco call out for pathogen survival in soil that we're hoping to be able to test for Pectobacterium as well. So, like John said, it is a needle in a haystack because there's probably in small amounts, but hopefully if that project comes to fruition, we'll be able to have another shot at trying to find any in soil.
Anybody else?
Ian, you'll remember some years ago we took samples from bad fields with a lot of blackleg and sent roots and shoots to you from weeds, and you did find blackleg bacteria.
So finding the bacteria is one thing. Knowing that roots are responsible for passing those bacteria onto potato crops is still something that's been very difficult to show. And actually, in terms of these bacteria on weeds, there are several countries around the world, and Finland and Israel did a particularly big study on this and found not just any weed, but certain types of weeds had these Pectobacterium and Dickia on them, but they still weren't able to categorically show that that was responsible for contamination on the tubers or blackleg in the field, although circumstantial evidence suggests that it probably is. So in terms of control, keeping weeds out of your potato crop would be an obvious thing to do in terms of trying to minimize this contamination, but very little in the way of proof. Okay, there's another question. Can verticillium in minitubers increase the risk of blackleg? And there's a follow-up. Previous studies have shown that almost 70% of minitubers tested do have that pathogen. And does the widespread nature of pathogens in general, and particularly verticillium within high-grade seed, need to be investigated? Is this something for you, Stuart?
I think it's more for Greg than me.
Okay, Greg, do you want to take that?
Right, wrong button. Yeah, it's a good question. Yeah, verticillium, it seems to be ubiquitous in all seed tubers, really. So we're not entirely sure if the connection between verticillium wilts and with blackleg is actually there. In terms of any disease, if it does enough to damage the plant, then yeah, there's definitely a potential for getting the bacteria in. We try to look at verticillium and blackleg sort of by co-inoculating the two diseases, but as I said, because it's verticillium so ubiquitous, it's really difficult to find any tubers that haven't got it. There is different species of verticillium that vary in their in disease that they cause, so it's something that could be looked at, yeah, and they do it to a limited extent in the minituber monitor that goes on at SASA. They don't find a lot of Verticillium dahlia, which is the main potato pathogen. They do find a little bit of Tricorpus and Alboatrum, which we don't think is an issue. So although there's a lot of Verticillium fungus in the tubers, it doesn't necessarily mean it's going to cause disease and then help Pectobacterium into the plant.
Okay, thanks, Greg. Anybody else want to comment?
Yeah, I think under warm growing conditions, it's probably shows up more with verticillium and other fungal diseases like rhizoctonia, and where you get these infections with fungal diseases, you tend to see more blackleg. That's just observation, really.
Okay, good question. Thanks, John. Okay, moving on. We've got a question here. moving seed from Scotland to southern England often happens when there's a big temperature difference between the two areas. What's the best way to handle seed delivered in these circumstances when it could have been in transit for more than a day? Stuart or Glyn?
Well, if the transit's in a day, there is likely to be the tubers will experience an increase in temperature as they move, as we showed from fairly short distance transport, and that could cause condensation. If the tubers, for example, left the store in Scotland at 4 degrees C and arrived in England at 12 degrees C, just to take an example temperatures, then yes, condensation could occur. So it does. Although the method of transport is pretty good these days and limits heat exchange a lot, there could be condensation, and that's why growers who receive seed should handle them immediately, ventilate and dry them immediately on receipt. Yeah, there is a risk there. One of the reasons for doing the experimentation was to see whether to try and explain why some people complained about blackleg in PB3, for example, yet the same grower growing the same seed on his own farm without transport was getting very little blackleg. So it does suggest that the length of time in a box or a bag and the conditions during transport could lead to increased Pectobacterium and therefore increased risk of blackleg.
Glyn, do you want to comment? Or?
I do. Well, I think Stuart showed the answer to the problem well in his presentation, and then the other thing was that the fact that you can get different levels of blackleg from the same stock in different fields as well, which might be one explanation for Stuart's observation of a grower. Yeah, so it's clear that you've really got to take care of the seed when you receive it and ventilating it, either by having it on a pallet and some form of even a plenum to create air through it, or moving it into a box.
Okay, thanks, Glyn. A question here: what are you planning to use to cover high-grade crops? That's definitely one for Greig or Stuart, because both have been involved in that work. Should we start with Greig?
So in terms of the project that Ian mentioned that we're currently started, there's a field trial that I'm hoping to start next year, which we would cover some of the crops with either polytunnels to keep out any aerosols and rain, and netting, so hopefully to allow the rainfall through and to keep insects out. So a combination of a field trial split into different zones that would be either open by polytunnels or covered by insect nets.
Okay, thanks. Stuart, have you got any comments on that? Anybody else?
Well, we did some work on covering PB crops some years ago, not very conclusively, but the principle is there to control the ingress of Pectobacterium into the crop. But it's quite an awkward thing to do, to grow mini tubers in polytunnels. Maybe a better solution is what some people are doing, and that's on susceptible varieties, home pulling by hand, and then avoiding the level of contamination you might get otherwise.
Okay, thanks. A question here, which I think is probably to me, although anybody's welcome to answer it. Have you isolated Pectobacterium atrosepticum within free-living nematodes? So we talked about the fact that if you add free-living nematodes to soil, you increase the ingress of bacteria into your plant up to 100 times. But I didn't mention what happens in terms of where the bacteria are. Are you just making holes that the bacteria can get into, for example, or are the nematodes actually carrying or vectoring the bacteria, or are the bacteria living on the outside of the coat? The answer to that is that we don't know, but as part of this new project, we're doing some microscopy that is going to help to answer these kind of questions. Are certain species doing something within this mixed population where they can vector the bacteria? Are the bacteria on the outside, or are they on the inside? Or are they completely unassociated with the bacteria, and the bacteria purely creating damage where they get in? So hopefully after another 2 or 3 years, we'll be able to answer that question rather well. So we look forward to doing that. Does anybody else have any comments on that?
I think it's definitely worth remembering that these bacteria are coliform organisms, and as such are natural gut inhabitants. So it would be not unexpected to find them in the guts of all kinds of organisms.
Thank you. A question here. Refrigerated stores are sealed boxes. Can the buildup of carbon dioxide affect PBA expression? We know it affects gangrene expression, for example.
John, do you want to start on that one?
So I think generally the amount of the CO2 buildup is, I don't know what levels they're talking about here. I think I'd be much more concerned with the sort of depletion of oxygen. So if the ventilation is relatively poor through the boxes, it's possible you might start to decrease the oxygen a little. And we know that this bacterium can cope, it's a facultative anaerobe, so it can manage in all circumstances. I wouldn't be too concerned, I don't think, if it was just going up to a few percent CO2, but otherwise the air was okay, and as long as the ventilation through the potatoes was good.
Okay. Thank you. We've got 2 more minutes, so I'll try and get a couple more questions in. Should you introduce heat into a store for drying crops? Glyn or Stuart, maybe?
Sorry, I didn't hear the question, Ian.
Should you introduce heat into a store to help dry crops?
Well, technically you can, as a way of warming the air up, and then increasing the amount of evaporation and air movement off the potato surface, because that dries it and slightly cools it. I say that if you've got any risk of soft rot or blackleg in your crop, I would be fantastically cautious about it. The prospect of having warm, damp crop is a risk.
Okay, thanks. Do you want to add anything, Stuart, or anybody?
No, very similar to that. Very similar to Glyn. I think I would be very, very concerned, especially if you were just trying to put the heat into one stock in a store with many stocks in. You risk condensation, and yeah, I think ventilation with outside air or with inside and outside air mixed is probably going to do you more good than putting heat in.
Okay, and final question. Is anything known about resistance mechanisms, with something we've not talked about today, that can be identified to enable molecular selection in breeding programs? Maybe I can take that one. We've done some work. We spent quite a lot of time doing work looking for signs, markers for resistance for PBA. I know other people have done it in the past, and it is proving so difficult to do. Almost certainly you don't get individual genes or individual markers for resistance. It's what we call multigenic, which means that lots of different genes are involved for different reasons in making some, probably not resistant. I don't think anything's truly resistant, but making them more tolerant. Some cultivars more tolerant than others. I suspect we aren't going to find good markers using conventional methods, and we're going to need to find new methods. For example, something like genomic selection, which we don't have time to talk about today, might help to answer that question. Or, dare I say it, something like biotechnology, which maybe in the future is going to be a possibility. So it's a good question, and the answer is it's very, very difficult to do and certainly not straightforward. And on that note, I'm going to stop the questions. I'm going to thank you all for coming, and I'm going to pass you over to Graham just to close the webinar. Over to you, Graham.
Brilliant. Thank you very much indeed, Ian. Could you move to the next slide, Christian? Firstly, big thank you to you guys for taking the time today to do this. Been way over my head, but I have learnt something, which is always a good thing to be able to say. And a big thank you to everybody who's dialed in and been sat watching this going forward. And I hope everybody has learnt something. Just need to point out that after the webinar, there will be a short survey that comes out. Please do take the time to fill this in. It's what we need to get the feedback, and then we know how to keep going for the future, because we will be doing more of these. Don't forget that your bases and Enroso points can be applied for if you haven't done so already. The webinar has been recorded and is available shortly online on the AHDB events page on the website, along with all of the webinars. And finally, just a quick plug for some really good webinars that are coming up through November. We can't avoid the old Brexit question coming in there. It's getting very close, and we've got a good one on the 12th about that. We can't afford COVID, so that's in there as well on the 11th, which is some really good insight into what's been going on. And we've got a one about soil on the 19th, which we've snappily titled "Why Bother?" It seems sometimes that we don't think the potatoes can have an effect, but they certainly can. And then leading up to a big day of events, the Potato Industry Conference with some international keynote speakers, so urge you not to miss that. Got the Aphids Day, and then we culminate with the Agronomy Week, which starts in November, but the potato section is more towards December. So finally, I think if you have any other questions or you want to get in touch, those are my details on the screen. We are also, as a team, we are all available via the website, all our different details for the different regions. So by all means, get in touch if you have any questions. There were a few questions we didn't get answered today. We will endeavour to get an answer out to people going forward. So it just goes to say, again, thank you very much to all the speakers. Big thank you to Christian in the background, my unsung hero, and the team for supporting. And most of all, thank you to you guys for attending. And hopefully we might see you in person sometime soon. You never know. So on that note, I'll say cheerio. Thank you very much indeed.